Hello, everybody. Welcome to my second seminar. Today I will discuss the aluminum toxicity and associated stress physiology in plants. So aluminum is the most abundant metallic element on Earth and the third most abundant element following oxygen and silicon. In fact, these elements are so abundant they combine to form aluminum silicate, which is the major inorganic component in clay soil. Shown here are beautiful aluminum silicate crystals, which gets weathered into alumina, silica, and eventually clay particle. Therefore, you can see that aluminum compounds is very abundant in soil and can make up some 20 to 50% of soil content. Normally, soil aluminum does not pose a threat to plants. This is because at neutral pH, aluminum is bound to silicate and oxygen. However, when soil becomes acidic, especially below pH 5.5, the solubility of these compounds increases, which releases the trivalent aluminum ion, or Al3+, which is the most dangerous heavy metal element in acidic soils. One reason being the general high abundance, the other one being the strong positive charge, which disrupts many biological processes, as we'll see later. Keep in mind, acidic soils also have other detrimental effects, such as nutrient leaching. Now, soil acidification is a natural process. However, it can be accelerated by human activities such as agriculture and fossil fuel pollution. And this has become a real problem around the world, threatening the global food security in the 21st century. As shown on the map right here, the most affected areas are the tropical and subtropical regions of the world. Now, these are some of the most productive lands on Earth and account for 50% of the total arable land mass. The results in yield loss due to aluminum stress can be as high as 80% in sensitive crop species such as wheat. All right, before we go further, here's an overview of the following discussions. We will first briefly examine the beneficial effects of aluminum to plants, then we'll focus on the phytotoxic effects, followed by some recent findings about the stress tolerance mechanisms. Lastly, we will address the biotech and scientific prospects. All right, now let's take a look at the beneficial effects, which is mainly manifested as growth stimulation. Note that aluminum is not an essential nutrient and has no known physiological roles in plants currently. So shown here in the left-hand side is the roots of tea plants. And as you can see, root biomass and length increases drastically when the plant is exposed to aluminum ions. On the right-hand side is a recent study in different rice cultivars. Similar to tea plants, the root growth is significantly higher when exposed to aluminum, and at the same time, the shoot growth is also enhanced. The rice plants exposed to the ion actually accumulated 140% higher biomass and double the amount of leaf chlorophyll contents. So these are indeed some very nice benefits. Of course, there's a catch. The beneficial effects only occurs at low aluminum concentrations and only manifested in plant species either native to acidic soil or especially tolerant to the ion. So shown here is a comparison of the differential effect of aluminum treatment in rice and barley seedlings. While root and shoot growth are enhanced in rice, as seen here in the longer roots, following aluminum treatment, the opposite is true for barley. As you can see, the growth is stunted at these concentrations. Indeed, rice is the most aluminum tolerant cereal crop species out there, and the tea plants we previously examined also has quite high tolerance. All right, now we'll get into the phytotoxic effect of aluminum ions. We'll first examine the symptoms and look at the cellular as well as molecular targets of aluminum. So the primary symptom of aluminum toxicity is root growth inhibition. This can occur rapidly within 30 minutes after being in contact with aluminum. Over time, the root would also become stubby and discolored, and extensive damage would occur. So as shown here on the right-hand side, the abnormal morphology of root apex in wheat and maize roots is the result of aluminum-induced damage. In the shoot, things starts to get tricky as the symptoms overlap with other nutrient deficiencies, making the diagnosis a bit harder. As you can see on the left-hand side, the leaf of ginseng and orange plants suffered from extensive chlorosis and leaf curling. At higher concentrations, there would also be necrotic spots on the leaf. So these symptoms are indeed characteristic of mineral nutrient deficiency, and we will see why that is later. Now, onto the targets of aluminum toxicity. Unlike other transition metals, unlike transition metals, aluminum is not a transition metal, such as iron and copper. Aluminum doesn't have any redox activities inside the cell. So the toxic effect is purely due to the strong positive charge. Keep in mind that most macromolecules are negatively charged in the cell. Therefore, aluminum really strongly and promiscuously interact with all of them. The uptake of aluminum occurs at root apex, where ions dissolves in the root mucilage and comes into contact with root epidermal cells. So this is demonstrated in this area here, where in the big red box are rice roots stained black for aluminum ions, and you can clearly see a gradient going from root apex towards the elongation zone, indicating the uptake starts at the apex. On the right here is an overview or list of the primary targets we'll talk about today, which is by no means exhaustive. So first, cell wall, plasma membrane, and symplastic transport. Then we'll talk about oxidative damage, mitochondria, and finally, nucleus. Okay, first of all, we have the cell wall. It is the major site of aluminum accumulation, which can trap up to 90% of the aluminum ion in some species. And the reason why cell wall really likes aluminum is because the pectin and hemicellulose contents. So you can see their structures shown here. And notice the exposed hydroxyl group on hemicellulose and the carboxyl group on pectin. So these sites contain oxygen, which is highly electronegative, and that really attracts aluminum ions. In fact, more so in pectin because it has, two oxygens. And here's the illustration of cell wall matrix, and aluminum binding to the hemicellulose and pectin really packs these components tighter due to the electrostatic interactions. As a result, the rigidity of cell wall increases. Moreover, aluminum can actually displace the calcium ions normally trapped in the cell wall matrices, which is needed for many processes such as acid growth. The combined effect is that root cell elongation is inhibited because of the high cell wall rigidity and ion imbalance. However, cell wall binding of aluminum can also be protective, especially in the less important cells, of course. This recent study right here showed that the root border cells, or RBC, can actually serve as aluminum sinks, which keeps the toxic ion away from the rest of the root tissue. During aluminum stress, the border cell would increase pectin synthesis, likely to enhance aluminum trapping abilities. So shown on the right here is the result of this study in Arabidopsis seedlings. So STAR1 mutants right here, they actually cannot upregulate pectin synthesis during aluminum stress. And as shown, right here, by the big red arrows, the aluminum ion stained purple really invaded the root tissue up to the elongation zone, while in the wild type, it's mostly trapped inside the root border cells. All right, now move on to plasma membrane. Remember, the phospholipid has exposed polar head groups, shown as the blue balls in this diagram. So these head groups consist of negatively charged phosphate groups and other components such as serine and choline, all of which attracts aluminum ions. Normally, the membrane is stabilized electrostatically by the calcium bridge between the head groups, and aluminum can actually come in and displace the calcium ions. As a result, there will be a stronger interaction between the lipids and head groups, leading to membrane rigidification, the loss of integrity. The binding of aluminum also contributes to endocytosis of this toxic ion into the cytosol. At the same time, the binding depolarizes the membrane, which hinders enzyme functions. So, speaking of enzyme functions, aluminum could directly interact with transmembrane proteins and inhibits their functions. As seen in the lower left region, the key plant enzyme proton ATPase, or HATPase activity, is significantly affected by high concentration of aluminum over time. The lower red bar represents the diminished enzyme activities at 24 hours into stress at this high concentration. So two effects immediately come to mind. First, this would hinder nutrient uptake from the cation exchange process from the soil particles, and the proton gradient driven secondary transport will also be compromised. At the same time, acid growth will be disrupted as well. Membrane-dependent signaling processes are also affected. As you can see on the right, aluminum ion inhibited IP3 production in tobacco protoplast in a dose-dependent manner, indicating the inactivation of phospholipase signaling pathways. IP3, of course, is an intermediate produced by the phospholipase. Note that PLC and PLD, so both phospholipase pathways, are important regulators of root growth. All right, symplastic transport is another major process that is affected. This is because aluminum binding to the membrane induces excessive callose deposition in the root cells. Remember from plant physiology course that callose is the stuff used to seal off damaged or infected vessel elements in xylem. And here it really blocks plasmodesmata in root cells, which abolishes symplastic transport. This is demonstrated in the electron micrograph of wheat root cells shown here. The little black dots are gold antibodies against callose. And you can see that in the red circles, the large amount of callose around plasmodesmata under aluminum stress really seals them up. This would, of course, disrupt symplastic transport, which is essential for sending water and mineral ions past the Casparian strip and to the shoot. By the way, this study right here directly showed for the first time that callose is deposited at plasmodesmata. So a quick recap of what we went through so far. Remember, the primary symptom of aluminum toxicity is root growth inhibition. So we have discussed how aluminum could rigidify cell wall, disrupt membrane-associated signaling and enzyme functions, as well as blocking plasmodesmata. These effects would inhibit root cell elongation and nutrient uptake. Coupled with the impaired nutrient transportation towards the shoot, we can now start to see why there would be signs of nutrient deficiency in the leaves. Of course, this diagram shown here is not the full picture, as we will see in the next few slides. All right, now we turn our attention towards the cytosolic side of the business. The most noticeable effect is the extensive oxidative damage experienced by the root cells. This is because aluminum ion can drastically increase reactive oxygen species, or ROS, production. Keep in mind, because of the unpaired electrons, ROS are extremely reactive and can irreversibly damage macromolecules. And these ROS come from principally two distinct sources inside the cell: mitochondria and plasma membrane. So shown here on the right are some common ROS encountered in biological systems. Okay, mitochondrial ROS. Now, once aluminum ion enters the cell, it can directly interact with electron transport chain, or ETC, proteins in the mitochondria. This would, of course, inactivate them, leading to a stagnation of electron flow, which provide opportunity for oxygen to come in and steal the electron and become superoxide. The superoxide would in turn generate a slew of other ROS. So the disrupted mitochondrial respiration can also lead to the depletion of cellular ATP and causing an energy crisis in the root cells. This would, of course, trigger necrotic cell death. Moreover, as you can see in these micrographs, the mitochondrial structure is severely disrupted by aluminum, as indicated by the red arrows. There are visible distortions to the crystallized structure and outer membrane structures. As a result, mitochondrial permeability transition pore would open and release calcium. This would activate the apoptotic cell death as well. So this study here gave us these nice visualizations of aluminum-induced mitochondrial damage. The other source of ROS is from membrane lipid peroxidation. Now, the ROS released from mitochondria or from general plant cell signaling can participate in iron-catalyzed Fenton-type reaction, which generates hydroxyl free radical. This can ignite a radical chain reaction within the membrane lipids. Remember, aluminum packs the phospholipids tighter together, which greatly facilitate this chain reaction. Shown here on the right is the MDA content in wheat seedlings treated with aluminum. MDA, of course, is a product of lipid peroxidation. Notice the amount of MDA significantly increases with increasing aluminum concentration. Similarly, up here, we have the wheat seedlings or wheat roots stained red for peroxide, and see at higher aluminum concentrations, the larger amount of peroxide present. Now onto the nucleus. By saying nucleus, it's really the DNA that gets targeted by aluminum. So if you look to the right on this diagram, aluminum is really attracted to the negative phosphate backbone of DNA molecules. The binding of aluminum would, of course, weaken the repulsion between the strands, thereby stabilizing the double helix structure, or rigidifying the DNA, if you will. This can disrupt regular gene transcription and DNA replications. The graph here demonstrates the reduced incorporation of radioactive thymidine nucleotide in tobacco protoplast. You can see that the DNA replication, right here, is drastically lowered in a dose-dependent manner, especially at 16 and 24 hours and with high aluminum concentrations. Note that the previously mentioned ROS can also enter nucleus and damage DNA. The combined effect of aluminum on nucleus is that cell division at root apex or root meristem stops, which inhibits root growth. Now, the inhibitory effect of aluminum ion on root cell division has long been thought to be caused by the increased DNA rigidity, which hinders double helix unwinding and replication. So that is actually not the case. And through a series of paper listed here, the mechanism is elucidated recently. All of these can be nicely summed up on a diagram to the right here. The inhibition of cell division is the result of activation of DNA damage response pathway in root cells, or DDR pathway, which causes the cell cycle arrest. This diagram looks kind of busy, but in essence, the WD40 protein respond to aluminum stress and recruit the ATR kinase, which activates many downstream targets such as transcription factors and other kinases. This would in turn, of course, upregulate DNA repair genes and cell cycle inhibitor genes such as the cyclin-dependent kinase inhibitors. Ultimately, these would result in cell cycle arrest and abolish cell division at root apex. And that is really all for our discussion with regard to stress physiology. Here is the previous summary diagram expanded with the new information added from the last few slides. This really highlights, it looks kind of overwhelming, but it really highlights a large number of processes affected by aluminum. So we have discussed the oxidative damage from mitochondria membrane, as well as cell cycle arrest that happens in the nucleus. Taken together, we can really start to see the symptoms observed in stressed plants. For instance, the root growth is inhibited from the disruption of both cell elongation and root cell proliferation. The perceived nutrient stress in the shoot arises from the impaired uptake, nutrient uptake and translocation. And finally, the extensive root damage and a barren morphology occurs as a result of oxidative stress in the root and extensive cell death. Keep in mind that there are still many affected physiological processes that are not discussed here today, such as the aluminum's effect on cytoskeletons. We merely covered a few of the most established mechanisms of aluminum toxicity. All right, now we'll briefly look at a few ways plants can deal with aluminum toxicity. The most popular tolerance mechanism is pretty much the same with any phytotoxic metals, which is from either exclusion or internal tolerance. So for exclusion of aluminum ion, the plants can produce a wide range of chelators to form complex with aluminum, which keeps it away from important macromolecules. The chelators come from many sources. For instance, the organic acids from Krebs cycle, such as citrate, malate, and oxalate. These ions would, of course, bind tightly with aluminum because they're anions in physiological pH. Secondary metabolites can also serve as chelators, such as hydroxamic acid and anthocyanin and other phenolics. So shown here are the wheat roots stained for aluminum and lipid peroxide. Notice the upper part separated by this big red line, where the anthocyanin accumulated and anthocyanin accumulated and no aluminum or peroxide accumulated. The lower part, especially the root apex, with no anthocyanin, really took a hit here from the peroxide and aluminum accumulation. So the chelated aluminum can then be exported into the apoplast or sequestered in the vacuole, of course, through a series of aluminum inducible transporters. And this diagram here sums up our discussion on the last slide nicely. So you can see that aluminum binds to cell wall and plasma membrane. Then when it enters the cytosol, it attacks mitochondria and nuclear DNA. However, it gets chelated by organic acids, which is not shown here, unfortunately. And the stable chelation complex is either secreted into the vacuole or excreted into the apoplast or soil particle by these ALMT1 or MATE1 transporters. Here, the ALS3 transporter can actually export the chelated aluminum into the shoot to kind of alleviate the toxicity in the roots. Now, moving on to the internal tolerance, there are also many different strategies that can be used by plants to reduce the damage done by aluminum. First, we have the methyl esterification of cell wall pectin. Remember, we discussed that aluminum loves the highly negative carboxyl groups on the pectin. Now, linking a methyl group to the exposed carboxyl group of pectin polymer reduces the accessibility and therefore the binding of aluminum ions. You can see here on the upper right-hand side the cross-section of rice roots. So this study used a fluorescent antibody against the unmethylated pectin. And notice how in the resistant strains of rice, there are much less unmethylated pectins. In other words, higher degree of pectin methylation is associated with increased aluminum tolerance in this case. Plasma membrane can also be modified to increase plant tolerance. Specifically, higher sterol content reduces the aluminum disruption to plasma membranes. So in this study right here, which is also done in rice, the resistant strain has a higher sterol content in the plasma membrane and accumulated less aluminum in the root apex as stained red. Here, applying the sterol synthesis inhibitors actually reversed this tolerance as shown in the red box here. And of course, plants can also produce more antioxidant to reduce the oxidative damage. We've seen this study before on the last slide. Now, phenolics such as anthocyanin not only serves as chelators, but also have antioxidant activity. Again, here, lipid peroxide only accumulates in regions lacking anthocyanin. All right, the last thing to discuss is the prospects. And we'll look at how the current knowledge can be applied in the field of biotech and agriculture. And then for the plant physiologists here, I'll address several key questions that needs to be answered. So first, let's see how aluminum stress is currently mitigated. The most common methods are various cultural practices. For example, you can raise the soil pH, which would reduce solubility of trivalent aluminum ions. This can be done with applying lime or gypsum. You can see on the picture right here, it comes in as a white powder. Another method is to use boron supplements in fertilizers. So boron is not only an essential nutrient for plants, it actually encourages the alkalinization of soil by plant roots. But this is, however, kind of inefficient because boron is a highly mobile nutrient and easily leached away. So you need a lot for it to work, really. Now, the most trendy method currently is to use biochar, which is, as the name suggests, charred biomass. So the biochar can provide a whole bunch of nutrients for plants, while specifically the silicon in biochar can form a complex with the aluminum ion and sequester it out of the soil. You can see here below in this paper, It's actually published just a few months ago in Nature. So indeed, the biochar is now very popular and is what, very trendy, very popular. Is what all the cool kids are using. However, while these methods discussed here are really effective, they start to get pretty expensive when we're using them for thousands of hectares of crops. And really, some of these are not accessible for developing countries such as Bangladesh. Therefore, generating aluminum-tolerant croplands is still an important endeavor. So how to increase plants' aluminum tolerance? There are many approaches, including the more obvious method, where you can just overexpress the chelator synthesis and transporter genes, which works out quite well here in this study. There are, of course, other ways. For example, in this study on the top right, they actually identified a transcription factor, Rinki47, in Arabidopsis, which serves as a master switch for cell wall content. So overexpressing Rinki47 reduced the cell wall pectin content and aluminum uptake, which increased the plant aluminum tolerance, of course. The other one we have previously looked at is the ATR kinase. So in the DDR pathway, actually not only the ATR kinase, but also other components in the DDR pathway as well. So attenuating their expression can help plants push through the growth inhibition caused by aluminum. However, one must be cautious to not reduce the DNA repair capabilities of the plants. And lastly, for plant physiologists here today, there are many critical questions that still lacks definitive answers. For example, we still don't know the specific mechanisms of how aluminum interact with various cellular components, such as the many transmembrane proteins. Another thing is that we still need to separate the primary targets of aluminum toxicity from the secondary effects. For example, we discussed the disruption of mitochondrial morphology due to aluminum binding. However, this can also be the secondary effect of other cellular stresses, which may also disrupt mitochondria structure. And we actually do not know that directly. The third and the biggest mystery, in my opinion, is the signal transduction involved in aluminum stress detection and response. Now, there are some evidence that suggests calcium, microRNA, and nitric oxide serve as the first wave signals when the root encounters aluminum. However, these are still quite controversial, and we don't really have a clear idea of the signaling processes and pathways involved. And that concludes my seminar. Thank you for listening.